exosomes. Actually, they are the usual secretory material from a cells, no matter if it's tumor cells or normal cells. Then certain genetic materials has been found to be packed inside exosome to be released by the tumor cells into circulation. We don't know what the message is because there is lots of message being packed in the exosome. It's just like these tumor cells or normal cells are sending off mails. And if you open the mails, it seems that there are different kinds of messages inside. And you can try to decipher the messages inside. Usually it contains quite a number of stable material inside. And for exosome, a lot of the times we find there is a package of microRNA inside. If this microRNA is not packaged in a mail, not packaged in exosome, if microRNA is being released just into the circulation, of course you can pick it up, but a lot of the times we believe microRNA is not stable enough to long lasting in the circulation for you to pick up consistently. So that's why we are interested in whether these mails or exosomes actually contain specific messages from the original cells telling what kind of cells in the distinct away from the circulation or in the same tumor. What's the message inside? And we don't know what's the exact messages to be found, but a lot of the times these could be related to metastasis. We don't know. And we have found that these serum exosomal microRNA profile could be assayed, and it seems that for patients with advanced stage non-small cell lung cancer, we can follow these serum microRNA, exosomal microRNA profile in a serial manner that some of them may correlate with the treatment response with stable disease and then turning into disease progression. We are still working on these panel of exosomal microRNA to see if it is related to the disease progress or to some other genetic materials or genetic process that we may not know. So for the circulating biomarkers, a lot of the times we need clinical validation, and whether it reflects tumor heterogeneity could be related to the underlying characteristics of the tumor, and it depends on whether we have good techniques to pick that small amount of biomarkers in the circulation. And for exosome, a lot of the times we need specific centrifugation, ultracentrifugation, in order to isolate the small amount of exosomal mRNA to be present in those exosomes. And hopefully we'll have these newer biomarkers for detection of early relapse or early stage disease. But unfortunately, right for the moment, I cannot think of any role of these biomarkers for the immunotherapy yet. Maybe for the future, especially, for example, on certain markers in the cytotoxic T cells. So today, hopefully, I have shared with you the application of these therapeutic biomarkers with EGFR, ALK, and also mentioned the immune checkpoint inhibitor, and also the our exploration into the circulatory biomarkers in advanced stage non-small cell lung cancer. And I do hope in the future I can share with you more, and we have further exchange with your unit in the future. Thank you very much.